Okay, hello everyone. We're going to get started. We were fiddling around with Zoom. There's some interest in other departments here. So welcome to the Hanover. It's the last Hanover part science seminar series, popular series for this semester. And I'm pleased to welcome Dr. Francesco Petruzzellis from the University of Padua in Italy. That's where I did my sabbatical last year, and so we worked together at world famous Ortal Botanical, which he'll talk a little bit about. Francesco is a research professor with expertise in plant functional traits. He studies drought, stress, plant-water relations, plant physiology, and he has a PhD in environmental life science from Università di Trieste.
Yeah.
And I'll just let Francesco.
Okay. Thank you very much for introducing me, David. Welcome, everybody. Good morning. So, as David mentioned, I'm Italian. I come from the city is Trieste, and right now I'm working in the city here, this municipality here, which is called Padua. And I am at the Department of Biology, but I'm super lucky since my office and my laboratory are not within the department building, but are placed in the botanical garden of the University of Padua, okay? So that I don't get all, the noisy meetings, annoying meetings, or it's usually that. So I'm really lucky since this is the oldest botanical garden in the world. It was founded in 1545. And we also have species that are really old. So for example, we have the species of Campcapsulis, Goethe's palm, which should be dated back in 1580, something like that. So in some decades, it is approaching 500 years old. Right? As David mentioned, I study functional trait. I'm in trait-based ecology, and I consider myself more of plant ecophysiology since I like going into the field, get dirty hands or wet hands in this case, okay? And I don't mind then get back to the laboratory and also perform measurement in order to extract all the possible information about the organisms that I wish to study. So let's say that the main character of my whole research activities are these guys here, the functional trait, okay? That are defined as any morphological, physiological, or phenological feature that can be measured from the cell to the whole organism level, okay? And these things that we can measure on plants must be linked to a function or to at least a set of functions, okay? And there is a very beautiful trick to explain to people that not used to work with functional trait, what is functional trait, okay? So let's say that the mostly used functional trait in trait-based ecology has been leaf mass per area, which is the ratio between the dry mass and the area of the leaf, okay? Sometimes it is expressed as specific leaf area, which is exactly the same, just it is the ratio between the area of the leaf and the dry mass, okay? So why this is so important? So just imagine that this is a leaf, okay? So we have a leaf with a certain area and with a certain dry mass, okay? So let's say that species with this kind of leaf have a very large leaf that intercept a lot of light, and so this kind of leaf can perform very high photosynthetic rate, okay? But then in nature, we can have species like this, okay? So here, the leaf area is half of before, so the light intercepted would be less, and so we can expect lower photosynthetic rates than the other. But if we look at the cost per unit area, okay, this will be double than before, okay? And we can go on, okay, and on until we get species with this kind of leaf, which are really tiny, really small, okay? So they will intercept, of course, a lot of more light than the first species, and so the photosynthetic rate would be lower. But on the other hand, the construction cost for the leaf area would be really, really high, okay? So why some species do have the first strategy and or this other second strategy, okay? So the thing is that if you try to break, okay, this kind of leaf, it will be really hard, okay? So this kind of leaf can resist better towards abiotic and biotic stress, okay? So that with just using one trait, okay, we can derive all this information, and we can actually classify species according to a gradient, let's say, of construction cost and photosynthetic rates. Okay, and this is not the only trait that we can measure, of course. On leaves, there are multiple traits, and in trait ecology, what we try to leverage are the the correlations among traits. Okay, and this correlation may reflect direct causal effect between traits, so mechanistic relationship between the two traits. Could be actually spurious correlation because there is a third or a fourth other variable that we're not considering that is correlated to both the trait that we are studying, or this is the most interesting, let's say, scenario, the most interesting kind of correlation that we are interested in, which is the coordination in trade-offs. Okay, so when I have to explain students what a trade-off is, okay, we don't have actually in Italian, we don't have a word that can have the same exact meaning that it is in English. Okay, so I usually show this picture here. Okay, we all see these movies here, and remember if this was the first Jurassic Park movies or the second or whatever, but we know that there are multiple errors in this movie. Okay, scientific-wise. Okay, and there is also this scene is so wrong. Okay, and because there is a trade-off that I will explain in a few more. Okay, so in this scene, we have people that are on a car and they are escaping from the T-Rex, the big dinosaurs, that is actually approaching the people on the car. Okay, but actually it is not possible since there is a trade-off between the maximum speed that the T-Rex could afford and then the pressure limit that can be then sustained by the skeleton of the T-Rex. Okay, so that these people here estimated that the maximum velocity of the T-Rex could be something in between 7 and 8 meters per second, okay, which is more or less 20-25 kilometers per hour. Okay, so it is true that the cars were electric and they were old, not the new brand electric cars. Okay, but I mean, okay, so we should. So in the future, if we have a massive attack from dinosaurs, okay, drive your car and you should be okay. So the same happens also in plants. Okay, so there are traits, functional traits that are coordinated with each other in trade-offs, and this is actually the most famous set of trade-offs that has been firstly introduced for plants, which is the leaf economic spectrum. Okay, so this was the first time that the authors leveraged online databases for which we had then data for hundreds of species of multiple functional traits. And they highlighted these two, let's say, mainly these two set of trade-offs which you see here. So remember, we were talking about leaf mass per area. And so higher values are related to species that invest more energy for leaf construction. And these species were the ones with the lower photosynthetic rate. So exactly as we hypothesized at the beginning, 2 minutes ago, okay? While on the other hand, we have species with very low construction costs, okay? And with very high photosynthetic rates, okay? And these species here are the one that you see here in this other graph, low construction costs are the one with also low leaf lifespan. So this trait here is leaf lifespan. The duration of the leaf is lower. And on the other hand, we have species with very high LMA and very high leaf lifespan, okay? So that with this three functional trait, they are coordinated in trade-offs that can tell us which is the strategy of the plant, okay, related to the resource allocation and to resource acquisition through photosynthesis. So that we can classify species along what we call the acquisitive conservative continuum, okay? So that resembling these trade-off here, okay? So this is the power of functional trait, and this is why functional trait became so used and so important in the last, let's say, 20, 30 years, okay? And 10 years ago, then this paper arrived. So it included much more species than the leaf economic spectrum and much more traits than the previous paper discussing about discussing about trade-offs, okay? So Here, the authors were able to retrieve for a lot of species, hundreds of thousands of species, 6 functional traits, okay, that are this one here with these labels. So, for example, nitrogen per unit mass, leaf area, seed mass, plant height, stand-specific density, and then leaf mass per area, okay? So, how to read this graph? So, when you look at this graph, the first thing that you think is about 2 fried eggs, okay? So, in response to fried eggs, okay? We can use the fried eggs, okay, to explain how to read the graph, okay? So, this is just an ordination, a PCA, okay? And each dot here represents a species, and the position of the species in this ordination depends on the value of the 6 functional traits that we measure here. So PCA are normally done in order to reduce the dimensionality and so increase the decrease the complexity of the database that you are using. Okay, so we have different colors here. Okay, and these colors depend on the density of the species present in each region of this ordination. We call it also functional space. Okay, so you see here, this is a similar ordination. We have again 2 axes, and here all the dots are species. And we can represent the position of the species, the probability of occurrence of a species with colors as represented here in this graph. So that for each region of this space, we can color each grid present, each cells present, imagine a lot of grids here, okay? In each cells, we can measure the actual probability density of occurrence of species in the functional space. So you see here that we have regions that in which we do not have any species, and this may represent regions where we have combination of traits that are not possible in nature, okay? Or we have here, for example, very bright colors, which represent regions where we have a high density, okay, of occurrence of species. And these kind of calculation falls under the framework of the trait probability density framework, which is now being used in order to retrieve a lot of information regarding the structure of the functional space, okay? So if you want to deepen, okay, there are some very interesting paper that tells you how to retrieve then indices that describe the structure of the functional space, okay? But going back to our ordination, here we have, as I told you, thousands of species, and this is the functional space representing the global spectrum of plant form and function. And we have multiple species, and we basically have 2 big regions with very high density probability of occurrence of species, okay? Meaning that we have a lot of species that have functional traits that follows, okay, these vectors here, meaning that here we have species which tend to have higher values of plant height, higher values of seed mass and leaf area. While on the very other hand of this space, we have species with lower values of plant height, seed mass, and leaf area. And in fact, on this hand here, we have species just as the Brazilian nuts, okay, which we ate the seeds. And on this very other hand of the functional space, we have species like Arabidopsis thaliana, okay, which is very short, with short leaves and also short seed mass, okay? So you see here that with just 6 traits, okay, we are now able to distinguish between herbaceous and tree species, for example, okay? And that based only on the relationship that exists among traits, okay? Again, the correlation among traits there can be assessed by calculating the angles between the vectors representing the traits, so that the closer is the angle to 0, the higher would be the correlation, the positive correlation among traits, okay? While the closer would be the angle to 180 degrees, okay? So we will have then a strong negative correlation among traits. If the angle is 90 degrees, like for example, almost between LA and LMA, we don't have correlation among traits. The 2 traits are independent of each other, okay? So the idea is that by calculating these spaces, we can reduce the complexity and so not consider all these traits separately each other, but we can actually calculate the component of the ordination so that we have the axis of variation that are somehow summarizing certain plant strategy. So in our case, for example, along the first axis, we can again have the leaf economic spectrum, and we can summarize our continuum of acquisitive and conservative species. Okay. So that was the starting point when I first started the PhD. And I had the feeling that was something missing, okay, in this, let's say, big equation, okay? So we had traits related to energy acquisition, nutrient acquisition and use, but nothing related to water, okay? So the 6 traits that we saw before are not related to how plants acquire water and then use water, okay? And so the idea at the beginning, when I started the PhD, was to include traits related to water use and acquisition into this big picture, okay? These kind of traits are normally deemed as hydraulic traits. And why they're really important, okay? So my professor of plant physiology, the first lesson told us that we don't have to seek for aliens outside Earth because we already have them here, okay? And these aliens are plants, okay? Since they do things that we cannot even think about it, okay? And water transport is actually one thing that it doesn't make sense, okay, for an animal, since plants are able to transport water from bottom to up, so in a different gradient, in an opposite gradient at the gravity force, okay, almost passively, so without using energy, okay? And this is because at the leaf level, okay, we have the transpiration process for which water is then lost in form of water vapor, and the transpiration acts as a force that pulls the water out from the plant and is transmitted through the trunk, through the xylem system to the root apparatus, okay? And the thing is that when we have stressful condition, so when the water availability is limiting, plants can die because of drought. They can dry out, okay? And to explain how it is possible, I put this glass here full of spritz. You know, in Italy, we have the already hours we drink Aperol spritz and so on, okay? And we can think as the Aperol spritz, okay, as the continuum of soil, roots, plant, and then atmosphere. In this sense, so the liquid in the glass represents the water in the soil, okay? The straw represents the xylem system where the water is transported in the plant. And then when we suck the spritz, the liquid out of the straw, this is the transpiration process that drives this water movement from the bottom to up, okay? So at the beginning, when we start drinking our cocktail, okay, we are all happy, nothing happens, the liquid is flowing and we are okay. But at a certain point, we start making strong noises, okay, unwanted noises, okay? And this is because air is entering into the straw and is making this noise. So the same happens also for plants. So when the water is limited, for example, in the soil, okay, so the tension that is then applied to the water column in the xylem system could be that high, okay, that then air bubbles are comes into the transport system in the xylem and then occupy this piping system instead of water. Okay, so that these water column here is being disrupted, and so plant is not then transporting water anymore. Okay, so there are critical levels of tension that plants can sustain in order to avoid, say, the formation of this emboli in the xylem system. And these thresholds are species specific, okay? And we can measure this threshold so that we can classify species in order, according to their drought tolerance, okay? According to their resistance towards the formation, for example, of emboli in the xylem system, okay? And to quantify the drought tolerance of plants, we basically use 2 main hydraulic parameters. The first is the water potential inducing 50% loss of hydraulic conductivity. This is a measure of the tension that the plant can sustain before losing 50% of hydraulic conductivity because of embolism information, okay? And on the other hand, we have the water potential, so a measurement of the tension in the leaf inducing target loss, okay? So if we consider the first trait, normally we measure this trait by measuring the so-called pressure volume curves with this very interesting instrument, which are called the pressure bombs, okay? Where we take leaves, okay? The leaves are dehydrated, okay? And then leaves are left dehydrated on the bench. And at certain dehydration level, we measure both the water content and the water potential using these instruments, okay? And we build these curves here for which you have a decrease in the water potential during the dehydration of the leaf, okay? At the same time, we can measure the target pressure in the same leaf, okay? So that when the leaf is dehydrating, the target pressure is going down, okay? And at a certain point, the target pressure will be zero, right? And this point, we can check the water potential. These points represent, okay, the pressure level, the water potential level inducing the target loss in the plant. This is important since if the target is lost, then the plant cannot perform photosynthesis anymore, and so on. And as you can see here, we have two species, Ola europea and Laurus nobilis, and these two species has different target loss point level. So for Laurus, the target loss point is minus 2.8 something, and for Ola europea is minus 3.5. Okay, so the lower is the value of the target loss point, the higher is the resistance towards drought conditions. And in fact, here is in this paper here, the authors were able to order the species living in different ecosystems according to their target loss point. And you see here that the value goes lower when moving from ecosystem where water is not a limiting factor to a more xeric environment. Okay. So the thing is that doing this measurement can be really time consuming, or you might have species like palms, for example, that has very large leaves and they cannot be fit into the instrument. Okay. So what we did is we developed a new framework, okay, a new method in order to retrieve this trait much faster. So I won't go into the detail. Okay, we can discuss more if you want later. But the thing is that we just need liquid nitrogen and the osmometer or dew point hygrometer, and we can measure the osmotic potential, the full target, and then retrieve the target loss point of the species. And we can speed up the process up to 10 times compared to the standard procedure with pressure volume curves. Okay, so that was perfect since now we have a method to measure hydraulic trait on a large number of species so that we can include this parameter in large analysis with a large set of species. Okay, we try to do also the same for the water potential inducing 50% loss of hydraulic conductivity, okay, the C50. And normally you measure this parameter by constructing the vulnerability curves. So again, you can have a leaf or a stem. It is fully hydrated so that the pressure in the xylem is zero. And then during the dehydration, the tension in the xylem decreases. Okay. And contemporary, you have embolism formation in the xylem so that during the dehydration, you have an increase in the embolism formation and so in the percent loss of conductivity. And from this curve, we can extract indices that can tell you whether a species is more or less resistant towards embolism formation. And one of the most used is the C50. Okay. And as you can see in this graph here, again, moving from areas where the water from more xeric area to more mesic area, okay, the value of the C50 is decreasing. Okay, so that with this just simple trait, okay, we can classify species according to their drought tolerance very easily. Okay, so also this method is really time consuming. So we try to do this with a very smart method, which I did not develop, okay, but I also try to use, which is the optical method. So the idea is that just imagine a leaf with veins, with xylem in the veins. So when the leaf is full tartar, the xylem in the veins is full of water, okay? And so that the light, if you put a light source over the leaf and you measure how much of the light is transmitted toward the leaf, okay, you will have a certain measurement. But when embolism occurs, okay, the xylem veins will be filled with air so that the amount of light that is transmitted will be different. Okay, and so if we subtract subsequent images, we can actually understand the exact point, okay, where we had embolism formation. So here, for example, you have an image of an ostracantinifolia, and you see that you have different colors represent different values of water potential where these veins, okay, embolize. Okay. So we did it using very simple equipment, so a very old smartphone, okay, and the LED stripe, okay, and we were able to measure the vulnerability curves for these two species, and this is the resulting patterns of embolism formation, okay, using this method, and the value of specificity was actually similar to the ones measured using standard hydraulic technique, okay. But still, with this method, you still have to wait for the leaf to dehydrate in the lab conditions. So for species that have, for example, very tight stomatal control, it could take a whole day or even more in order to retrieve very low water potential. So we tried also by injecting the leaves with air in order to rapidly induce the embolism in the leaf. Okay, so for example, you could measure an entire vulnerability curve in half an hour compared to 1 day, for example. And but we ended up in having for some species values that were quite higher than the ones calculated with the standard technique. Okay, and it seems that species with very long vessels, okay, in this species you have the highest bias. Okay, so that we still do not have a framework that can allow us to have a very super fast quantification of xylem embolism. Okay, so for the next analysis, for the analysis I did then later in my PhD, I just had to use the Targolos point. Okay, but I'm still trying to figure out how to retrieve also this parameter here in a faster way. Okay, and so during my PhD, I started using this trait to answer to let's say more ecological sound questions. So, for example, which was the role of hydraulic traits in shaping plant invasion? Okay, so we know that invasive plants are enormous problems worldwide. They have strong impacts on biodiversity. So the reduction of biodiversity is often associated to the increase of the presence of invasive species, and we have direct effects on the economy. For example, just by the management of the species, or indirect effect because of detrimental effects on human health. Okay, so that understanding which are the traits that enhance or that makes an island species invasive could be important in order to understand how to deal with these species. And so I did analyze different invasive and native species in our regions. So one of the main invasive tree in Italy and the whole Europe is Ailanthus altissima, the tree of heaven, which is invasive here in the US, right? And so as first analysis, I compared the functional trait of the tree of heaven with its native competitor, which is Fraxinus ornus in our latitude. Okay, and I focus on hydraulic traits, and what I found is that Ailanthus altissima has lower safety. Here you have lower values of Targolos point, lower values of C50 if compared to Fraxinus ornus, but at the same time it has very high water transport efficiency, but really, really very high compared to Fraxinus ornus. Okay, so then I upscale this kind of analysis, including more invasive and native species. So I sampled in 6 different sites that differed accordingly to their water availability. So I had both coastal dunes, for example, very arid site, grasslands, and sites nearby lakes and rivers. And I sampled 93 species, 15 woody, 70 herbs, and 15 invasive and 78 invasive. Okay, and I measure a bunch of functional traits, okay, that are related to say these axis of variation of the functional space. So I measure traits related to leaf construction cost, like for example the leaf muscular area that we saw before, and then I measure hydraulic traits like Targolos point related to the vulnerability to drought. And then I also measure the vein density of the species, which is mechanistically related to the hydraulic conductance in it, so that could be considered a proxy of water transport efficiency. Okay, and what I found, I calculated the functional space resulting from this survey, and you have the same functional space. Here you have in green the probability density of occurrence of native species, and here the ones of invasive species. And as you can see here, while native species, they have many different strategies, okay, possibly accordingly to the site where I sampled this native species. Invasive species tend to occupy only this region of the functional space, indicating that they have very specific strategies, and they share these strategies even if these species comes from different parts of the world. Okay, so for example, in this set of species, I also had Amorpha fruticosa and Robinia pseudacacia, which are native from North America. Okay, and they had similar strategy than Ailanthus altissima, which is coming from Asia. Okay, so by calculating again the trade-offs between traits, what I ended up in highlighting is that also species with lower construction costs had lower resistance to drought and higher water transport efficiency. And the invasive species tends to be at the very at this end of the spectrum, while native species tend to occupy the other end of this spectrum. Okay? And then also that the lower resistance to drought was associated to higher water transport efficiency. Okay, and you have and again you have a clear separation between native and invasive species. Okay, so that invasive species had very low resistance to drought. They had very risky strategy. Okay, but the benefit is that they have a very high water transport efficiency. Okay, and here you have a glimpse of the different venation density of one invasive species in Europe, Amorpha fruticosa and Francesco. The magnification is the same in these two images. Okay. So yeah, this is what I ended up with. At lower latitude, okay, the strategy that invasive species that seems to be successful for alien species in order to be invasive is to have very high leaf level water transport efficiency, implying a lower safety to drought, but with the advantage of reduced cost for leaves and wood production. Okay, so that next time we want to import a new species, we might just measure this functional trait, okay, so that we can understand before if the species can become invasive or not. Okay, but then calculating all this trade-off led to the idea that maybe hydraulic traits are telling us something that is a bit different than the things that the leaf economic spectrum and other traits are telling us. Okay, so I increased this database just with herbaceous species at the moment, and then I calculated the functional space resulting from all the traits that I already mentioned, and I calculated the minimum number of components, the minimum number of axes that have to be retrieved in order to describe the functional space of this set of species. And it ended up in having three significant components that should be retrieved. Okay, and the first component seems to be the ones related to the leaf economic spectrum. Here you see these are the correlation coefficient of each trait to each component. So this is the first component, and these are the highest correlation coefficient measured, and these are associated to traits of the leaf economic spectrum. Okay, then we have a second axis of variations, which seem to be related to the vein density trait, so to the hydraulic efficiency of the leaves. And then we have a third axis that is only determined by the Targolos point, so the axis related to drought resistance. So this analysis suggests that hydraulic traits are independent from the leaf economic spectrum, and also that hydraulic efficiency and resistance could be independent from each other, and that by measuring this trait, we can capture three main axes of variation of plant. Okay, so that we can increase let's say, the complexity and our understanding of the functional space. Okay, and we can use this information to describe for example, to monitor the diversity over space and time, so that we can measure this trait and calculate indices of functional diversity based on the calculation of the functional space. Okay. But the thing is that sometimes it is really hard to upscale from the level of individual to the community. Since to measure traits, you still have to go into the field, collect samples, go back in the lab and measure the samples. So that if you want to do to have this information over a large spatial and temporal scales, it might not be feasible to use a field approach. Okay, but thanks to us, a new era that's right, not right, it's rising, yeah. It is the era of remote sensing. Okay, so that the idea is that we can use the modality of how light is reflected by vegetation in order to retrieve these functional traits. Okay, so here you have just a simple example of this is a spectral signature of the species. So this is the reflectance values, the typical reflectance values of a plant in the solar spectrum. Okay, so ranging from 400 to 2,500 nanometers. Okay, so if you see, for example, here we have two species. So this is corn, maize, and this is the potato, Solanum species. And you can see here in the black line, you have the reflectance values of the two species. And as you can see, especially in this region here, the two species have different values of reflectance. So implying that they reflect light in a different way. Okay. So the idea is that we can retrieve these differences and use this difference in order to predict and model different functional traits. Okay. And we are now in the process in understanding which functional trait can be predicted according to which region of the electromagnetic spectrum that we are considering. Okay, and so this is, let's say, the path that my research activities are is taking. Okay, I'm just showing this simple study here that we did in a coastal ecosystem where we were interested in relating the indices of functional diversity retrieved with the field sampling campaign with indices of spectral diversity retrieved using a drone equipped with a multispectral sensor, okay, that has 5 different bands, so that can acquire the reflectance in these different regions of the electromagnetic spectrum. Okay, so we calculated the functional and the spectral space using the same approach, so using the calculation of this probability density of occurrence. Okay, and this is the functional space resulting from functional trait, and this is the resulting spectral space. And then we apply some statistical tools, I won't go into the details, in order to understand whether the different plots where we sample there, whether the spectral and functional diversity were related to each other. Okay, and what we ended up is that they are correlated indeed. And how to read this graph? Here you have increasing dissimilarity in spectral diversity among plots, and here you have increasing functional dissimilarity among plots. Okay, and so in 6 different transects, we found always this positive correlation, meaning that the higher is the diversity of strategies of the species present in the plot, the higher would be also the diversity in the spectral response acquired, spectral signature, sorry, measured with the drone. Okay, so that this might indicate that we can use this technology in order to quantify the functional diversity present in a certain area. Okay, so I try to do this by upscaling this measurement up to let's say a higher scale. So this is Friuli-Venezia Giulia region, which is actually the region in Italy where I come from. Here is Trieste. It is in this point here. Padua is here. Okay, and so we take advantage of the Italian National Forest Inventory, so that we have multiple plots in the mountain regions in Philippine and Nigeria, and for each plot we had the abundance of each species, the 3 species present there, and we retrieved functional traits both from online databases, and also we went into the field collecting again these samples for the species for which we didn't find traits in the literature. Okay, and as spectral data, we used the reflectance values coming from the bands of the Sentinel-2 satellite, which is a satellite that continuously orbits around Earth and is measuring the reflectance in this band here. Okay, and we applied the same approach that we applied before. We were first interested in understanding whether we can predict single functional trait using the spectral reflectance from Sentinel-2, and then also to test again the relationship among indices of functional and spectral diversity. Okay, so what we found is that there are some traits that could be predicted, okay, using the spectral data and also the data coming from online databases, while for other traits this is more trickier. So for example, for the C50, the accuracy level is that low, so that you can conclude that we are not able to predict this trait with a good accuracy. Okay. Then again, we calculated the functional and spectral space. Okay. We tested whether which was the structure of these spaces. So regarding the functional space, we had a clear separation, okay, of the plots where we had a higher abundance on gymnosperm species, in contrast with plots where we had a higher abundance in angiosperm species. Okay, of course, these 2 kinds of plants are adopting a very different strategies, at least at our latitude. Okay, so we calculated then that indices of functional and spectral diversity, but what we found is that the relationship was not that high as in the case study of coastal dunes. Okay, and you have let's say an idea of what we found in our analysis. So here we have, this is the plot number. Okay, this is the composition of the plots. So in all these plots, we basically have 2 species, Fagus sylvatica and Pichia abies, and these 3 plots have the same species, but the abundance of these species is different. Okay, so you see here, for example, the abundance is similar between the 2 species. Here we have the higher abundance of the gymnosperm, and here the higher abundance for the angiosperm species. Okay, and as you can see, actually the functional space, so the this index, functional richness, is a proxy of the volume of the functional space occupied by the plot. The value is the same, okay, since we only have 2 species with the same trait in each plot. Okay, but the spectral space was much different. Okay, so that when the abundance was close to the 2 set of species, we had a lower volume of the spectral space occupied, while when the abundance changed, also the volume of the spectral space changed. Okay, and this led us to hypothesize that the stem structure, okay, and so the structure of the tree might have an impact on the spectral reflectance that is measured by satellites, for example, and so that we somehow have to find a way to quantify this effect and try to understand whether there is also a relationship within the functional space. And it is actually something that me and David want to search over with our collaboration. Okay. Yeah, and this is the conclusion. So let's say that in the future, the idea is that I still want to continue to develop rapid and reliable frameworks to increase our ability to measure hydraulic traits. And then the idea is that we have to increase the set of species in order to understand whether hydraulic traits are or not coordinated with traits, for example, of the leaf economic spectrum. And the botanical garden of Padua is perfect since we have species from many different biomes, so with very different strategies, so that I can access to, let's say, these this set of species really easily. Okay, and at the same time, I am constructing a lab where I want to have many different multispectral and hyperspectral sensors in order to understand whether it is possible and how good could be the accuracy of the estimation of functional trait at different spatial scales. Okay. So thank you very much for listening to me, and if you have questions, I'm here. Okay.
So we'll take, Francesco will take questions from the room, and then also if Aki or someone else is monitoring, we can see if there's anyone who has questions on Zoom as well. But why don't we start in the room first, or we figure out the Zoom thing? Someone. I wanted to start with, so in the earlier, you talked about implications, and I thought it was interesting, there was some study about water use. It seems like one of the implications was that invasive species suck a lot of water out of the ground. So I can't remember the species, but two species that had invaded the Florida Everglades. There was a big concern. Melissa, that it was actually lowering, it was actually draining them through this high water use efficiency. So I thought that was a fascinating, conclusion in terms of the implications of this.
Yeah.
If you want to comment more on that, or if people have looked further into that aspect.
Yeah, there are people that went further into this aspect, and the thing is that they mainly try to analyze global data set, but of course the strategies that allow alien species to become invasive are context dependent. Okay, so this is what I found for my latitude. Okay, in Europe, this may be something in some regions here in the US. But when you try to test whether it is happening at any kind of context, you might find some contrasting patterns. Okay, so it seems that it is not a trait that is shared among all the species that can possibly be invasive, but there are some regions, okay, where this trait is really something. Okay, and it is curious since these very risky strategies is adopted by species that are invading medium xeric environments like the ones in the Karst region where I come from, okay, where we have strong periods with low water availability and high temperature. Okay, so for example, Ailanthus altissima has a very long rooting apparatus, so the root can grow up even 50 meters from the mother tree. Okay, so the idea is that it can access to many different ecological niches. Okay, and so this is a way in which it can outcompete plants. And then one thing that I did not consider here is also the phenology of the plant. Okay, so it might be that the water exploitation, okay, done by invasive species is done very early in the growing season rather than down in the growth period. Okay, but this is something that should be tested. Yeah.
Yeah, I was just wondering if you could comment a little bit on that finding of, I guess it's a bit orthogonal, the xylem traits versus the water use efficiency traits. And I understood as being more peak focused. Not sure if I understood that correctly. And is that similar to what other folks have found? I know there's sort of like, you think there's kind of a conductance versus resilience trade-off that can, I guess, be broken or is flexible in a few ways. So I don't know if that's surprising or what you'd expect or what.
Yeah, so again, yes, most of the data are, all of the data that are presented are leaf data, right? Mm-hmm. It's easier to make a leaf trade on leaf, okay? Yeah. And still, for drought resistant, for stem trait, we still do not have this very fast and reliable trait, okay? And yes, regarding the trade-off between safety and efficiency, product safety and efficiency water transport. So there is this seminal paper where they found that there is not a clear trade-off, or as we call it, a mechanistic trade-off. So that you expect a linear regression and all the points in this line. Okay, but what they found is that there is there are combination of these two traits that are not possible. So for example, you cannot find species with very high resistance and also with very high water transport efficiency. Also, there are some complaints regarding the method used for measuring these traits. So for C50, there are a bunch of methods ranging from classic hydraulic or X-ray based, synchrotron based X-rays, or rotors, and there are still people are still fighting about which is the best method to do that. But the thing is that still, when you reduce the set of species to, of course, the invasive species that are present in the latitude in Europe, it seems that this trade-off can explain, okay, the distinction between native and invasive species. Okay, so it might be something to consider when trying to understand why the nine species is becoming invasive. Okay.
Can I just ask a short follow-up? Any connection with stomatal regulation?
Yeah, I didn't measure actually, for example, gas exchange rate, even because all these traits are more pattern traits. Okay, so LNA, when the leaf is mature, it doesn't change. While gas exchange can have can change. But the thing is that, for example, for Ailanthus altissima, this is a species with very high tomato control. Okay, so that when there is some drop event, the species can close immediately stomata and so can save water for later. Yeah, and I know that there are people that measure, for example, the oxygen isotopic composition of invasive species and to understand and they found some partners, but the professor should be Moni Cavalier. But after a few papers, I didn't find any other papers on that. But it would be nice if there was data.
Scott asked it.
Okay.
Thank you for converting. Is there anyone online?
I don't see everything.
Okay.
You had beat me to it originally, but I did really appreciate the consideration of evaluating hydraulic traits as a potential aspect for evaluating the invasiveness of any potential imported plants. Do you think there are any other traits or aspects of the plant that you think broadly would be good to evaluate for imports to determine its risk before we bring it in?
Yeah, sure. Reproductive traits, for example. So I didn't measure any, but of course, the number of seeds simply could be also a good proxy, okay, for deciding whether or not to import the species. So, for example, Ailanthus altissima, actually it is a dioecious species. So the females, when the seeds that they can produce can be 10, 20,000 per plant. Okay, so a lot of them. Okay, so that would be something to consider. So reproductive trait.
Thank you.
You're welcome.
Can I ask another question? Sorry to just want to ask you about climate change. I get the sense, thinking about invasive species, we do see this general pattern that you have these faster growing invasive. And I mean, a lot of that's got to be up to taking advantage of more disturbed niches, right? I mean, in the broadest scope, right? And then there's traits associated with that. Do you think that these are traits that end up being less resilient in a climate change world, especially when you're in semi-arid systems? So you wonder if you see a sort of ecosystem consequence where now some of the invasives aren't doing so well in droughts, or if it just means that there's a bigger sort of flash of productivity and bigger impacts of droughts. What's your take on that?
Yeah, it's a very interesting question, and we wonder that whether right now we are living in certain conditions, and so this kind of strategy is successful. But what happens if the condition worsen, and so this very risky strategy led to the one that okay. So we did some specific experiments on Ailanthus altissima and Asia, trying to measure how good they are in recovering them the hydraulic transport after drought. Okay. And it seems that Ailanthus altissima is really good in doing that. Okay, and we're still trying to figure out whether it depends on some process like the refilling process of the embolized vessels and so on. So this plant has this very large peak okay, and a lot of parenchyma stems so that you can store a lot of water there, also in the roots, and you can explore. We don't know actually, okay, where the roots of the species are, okay, so that yeah, it would be nice to have more data on that, trying to understand which could be the maximum level or the maximum change that the species can support in order to understand whether in the near future we will have like a desert, you know,
Well, the whole world's a desert, we might not care anymore.
yeah, you're right.
On that happy note, so we have, if you signed up for lunch, we have lunch downstairs, and we can continue the conversation with Francesco. Thanks again for a really interesting presentation.
Thank you. Thank you very much.